Dear students, welcome to 12th class Biobotany. So we were discussing about the chapter Asexual and Sexual Reproduction. In this chapter, we already covered asexual reproduction in plants, that is vegetative propagation, and natural methods of vegetative propagation and artificial methods of vegetative propagation, and also conventional methods like cutting, layering and grafting already we have covered. And one topic left, what is the modern methods of vegetative propagation. Okay, so we are going to discuss about modern methods of vegetative propagation. In year 1932, Professor E.C. Stewart, he did an experiment. He took flowering parenchyma cells of carrot plant and he put all these flowering parenchyma cells in a culture medium. Suppose this is the carrot plant. He took the carrot plant flowering parenchyma cells and he put them in a culture medium under controlled condition. Then he observed that these cells, they divide and multiply and they become developed into a complete new carrot plant. Okay, so by this experiment what you can understood that plant cell, they have a ability to develop into a complete plant or entire plant under controlled condition. Plant cell, they have a ability to develop into a complete plant under control condition. That ability of plant cell is called totipotency. Okay, so what is totipotency? The genetic ability of plant cell to produce entire plant under suitable control condition. So here you see a carrot plant. We took the carrot plant cell and we put them in culture medium, and also we maintain control condition. We have provided sterile condition and we have given hormone treatment. Then after what you observe, these cells, they divide and multiply, they become into a complete plant. Okay, so what is the ability behind this result? Totipotency of the plant cell. Okay, and this totipotency we have applied in tissue culture. So by tissue culture we develop many plants. Okay, and we try to propagate the plants that produced by tissue culture. That is called micropropagation. Okay, so a modern method of vegetative propagation is called micropropagation. The tissue cultured plants we use it to cultivate. Okay, that is called micropropagation. So micropropagation means here we developed the plants from tissues or cells or maybe vegetative parts, maybe from stem or leaf or root, into a complete plant through tissue culture. That is called micropropagation. So what is micropropagation? We developed a complete plant from tissue or cell or vegetative parts of the plant through tissue culture, and we used them for cultivation. Okay, that is called micropropagation. So micropropagation is widely used modern method of vegetative propagation. Okay, so in agriculture field and horticulture field and also in industrial field, this is the way of propagation of plants, that is micro propagation. We can develop many plants in a short period of time by micro propagation, because here we are using only plant tissues or cells, and we put them in a controlled condition or sterile condition. Then we gave them hormone treatment, other nutrient medium. Then after some days we can observe they develop into complete plant. Okay, and also we get genetically identical plants, disease free plants. Okay, so these are the advantages of micropropagation. We will discuss what are the advantages and disadvantages of micropropagation. So let's discuss what are the advantages and disadvantages of micropropagation. First of all, first advantage, we can multiply the plants in rapidly in a short time. We can make many copies of the plants in a short time. That is the first advantage. And second advantage, the plants which are produced by micropropagation, which are genetically identical. Okay. Then third point, the tissue culture or micropropagation we can perform at any season, whatever it is, summer or rainy season, we can perform at any season. Then endangered plants can be propagated by micropropagation. Endangered plants means those plants they are facing extinction, they are disappearing from this earth. Okay, so those plants we can propagate it by micro propagation. And also meristem culture, it is a type of micro propagation by which we can develop disease free plants, because meristem cells, the plant virus which cannot infect meristem cells, because meristem cells are actively dividing cells. Okay, and if you take meristem cell and culture those cells, and we can develop disease free plants, that is called meristem culture. So by meristem culture you can develop disease free plants. So these are the advantages. And what are the disadvantages? In this micropropagation we need we need skill, and also labor cost is very high, and also we have to maintain controlled condition or sterile condition for performing micropropagation or tissue culture. And also while performing this micropropagation, the callus, that it means the developing plant tissue, is called the callus. Those callus may bear some undesirable characters. Those undesirable characters may be economically not useful, so we may get the plants with less economic useful. So that is also disadvantages of micro propagation. Okay. Next we are going to discuss sexual reproduction. So what is mean by sexual reproduction? Sexual reproduction means male gamete and female gamete, they fuse and they develop into a new organism or new plant, that is called sexual reproduction in case of plants. Okay, so there are two steps in sexual reproduction. First one, gametogenesis, and second one, fertilization. What is mean by gametogenesis? Gametogenesis means development of gametes or formation of male and female gametes is called gametogenesis. Formation of male and female gametes. Then what is fertilization? These male and female gamete, they fuse and they develop zygote, and they divide and multiply into a complete new individual. They form a new individual. Okay, or complete plant. So that's about sexual reproduction. So sexual reproduction, there are two processes. Gametogenesis, that means formation of male and female gamete. Then second one, fertilization, fusion of this male and female gamete, and they will develop into a new offspring or new children. Okay, so in case of plants, you know, flower is the reproductive organ. Flower is the reproductive organ, and which is a modified shoot actually. So in +1 we already studied different parts of the flower. You know that they have 4 whorls. Flower, they have 4 whorls. Calyx. These whorls are called calyx. They consist of sepals. So sepals, they form calyx. Then after calyx, corolla. So these are corolla, and corolla consists of petals. So petals, they form corolla. Okay. Then androecium, the male part of the flower is called androecium, and which consists of stamens. So these are stamens, they form androecium. Then gynoecium is the female part. Gynoecium, which consists of carpel. Okay, this calyx and corolla, they are called non-essential whorls because they have no role in reproduction. But androecium and gynoecium, they are essential whorls of flower. Essential whorl, because they are involved in reproduction. Okay, so this is about flower. So what are the different parts of the flower? Calyx, which are made up of sepals, and corolla, which are made up of petals, and androecium, which are made up of stamen, and gynoecium, which are made up of carpels. So androecium, which are made up of stamen. So what is the structure of stamen? Stamen, they have a long, they have a stalk-like structure that is called filament, and they have a bag-like structure called anther. This is our stamen. Okay, filament which is attached with anther. Inside this anther, what present? Pollen grains present. Pollen grain. So this is stamen. Stamens, they form androecium, the male part of the flower. Then what about the gynoecium? Gynoecium, which consists of carpel. So this is the structure of carpel. At the top, they have a knob-like structure, that is stigma. The anther from the pollen grain from one flower which land on this stigma, and they germinate on this stigma. Okay, so pollen grain, they land on the stigma of the carpel. Then they have a long tube-like structure that is called style, and this style finally lead to ovary. And inside this ovary, what form? Ovule form. Ovule. So this is general introduction about structure of flower. They have calyx and corolla, and also androecium and gynoecium. Okay. So sexual reproduction in plants takes place by three stages. First one, pre-fertilization events. Second stage, fertilization events, and third stage, post-fertilization events. Okay, so you can observe at the time of sexual reproduction in plants, you can observe pre-fertilization changes and fertilization changes and post-fertilization changes in plants. So now we are going to study what are the pre-fertilization changes takes place in higher plant during the time of sexual reproduction. So you know, we already studied flower, how this flower form. The flower, they formed from the flower primordium. Okay, this flower primordium, these are group of cells, they converted into flower by the hormonal or structural changes. Then they develop corolla, calyx, corolla, androecium and gynoecium, etc. Androecium and gynoecium, they are essential words and they are involved in reproduction. So first we are going to study about androecium. Androecium, we already know, the androecium consists of stamen which have filament and anther. And inside this anther, what present? Pollen grains present. And pollen grains, they are called male gametophyte because the generative cells of pollen grain which divide into male gametes. Okay, so that structure which lead to production of male gamete, it is called male gametophyte. So pollen grain, it is a male gametophyte. The gamete producing stage, that is pollen grain. So how this anther, the pollen grains are found in this anther. How this anther developed at the time of sexual reproduction, that we are going to study. First of all, you can see at the time of anther development, the anther primordium, that means a mass of cells and a mass of cell which are surrounded by epidermis, that is called anther primordium, is formed. What is first step of the anther development? A mass of cells which are surrounded by epidermis, that is anther primordium, it is developed. Okay, and this anther primordium, or is a mass of cells of anther which continue its development and it become like a four-lobed structure. Okay, and this four-lobed structure, what happens? Some cells of hypodermal cells, this is epidermis, and these are the cells found below the epidermis, it is called hypodermis. Some cells of hypodermis which become enlarged with a conspicuous nuclei. So these hypodermal cells which become archisporial or archisporium or archisporious cells. So the hypodermal cells which become enlarged and become archisporium cells at each lobe of anther. So this is archisporium at one lobe, and this is another in another lobe, and this is another in another lobe. This is one in another lobe. Okay. So archisporium cells, archisporial cells are developed. So how they are developed? The hypodermal cells which are converted into archisporial cells. Then what happens? This archisporium which is differentiated, they undergo periclinal division. What is meant by periclinal division? Just notice, that means parallel division of cell is called periclinal division. This is the one cell, and that cell divide parallelly and produce two cells, that is called periclinal division. This archisporial cell, they undergo periclinal division and they form parietal cells and sporogenous cells. Okay, suppose this is our archisporium cell which undergo periclinal division. They form parietal cell and sporogenous cells respectively. Parietal cell towards epidermis and sporogenous cells to inner side. Okay, and this parietal cell you can see here in this lobe, parietal cell here and sporogenous cell, parietal cell, sporogenous cells. Same here, parietal cell and sporogenous cell, parietal cell and sporogenous cell. Okay, and this parietal cell which undergo again anticlinal division and periclinal division. Anticlinal division means the cell which divide right angle to right angle direction. So that is called anticlinal division. So this parietal cell which divide periclinally and anticlinally and form 2 to 5 layers of cells, that is called anther walls. Okay, anther walls are developed. So this parietal cell, they undergo periclinal division and anticlinal division, they form anther walls. Okay, so what are the anther walls? Like first of all endothelium, middle layer and tapetum. All these walls are developed by parietal cell division. Understood? Then what happens? We have sporogenous cells remain here. Okay, these sporogenous cells which undergo meiotic cell division. Sporogenous cell. Sporogenous cell undergo mitotic division. Mitotic division, and which form microspore mother cell. Microspore. Okay, so they will develop microspore mother cell here. So I didn't draw that picture. You look into your textbook. So sporogenous cell which undergo mitotic division, they develop into diploid microspore mother cell, and this microspore mother cell undergo meiotic division, and finally they form microspore. Microspore. Okay, so that formation of microspore from microspore mother cell is called microsporogenesis. So these are the steps during the time of anther development. We describe in very detail how anther development takes place. Okay, so finally just a review how anther development takes place. First of all, mass of cells are developed, in which hypodermal cells, they are hypodermal cells which become archisporial cells, and these archisporial cells undergo periclinal division which form parietal cell and sporogenous cells, and this parietal cell undergo periclinal and anticlinal division which form anther wall layers, endothelium, middle layers and tapetum. What about sporogenous cells? They undergo mitotic division. They form microspore mother cell which is diploid cell, and this diploid microspore mother cell again divide by meiotic division. They form microspore tetra. That means that is haploid cell. Microspores are haploid. They form like a tetra. They stand together like a tetra. So this is one microspore, this another one, and this another one, and this is another one. So they form a tetra like, and this from this tetra, each microspore they free and they converted into pollen grain. Okay. Then in some higher plants, the microspores present in microsporangium or in anther, they held together like a mass. The all microspores they held together and they form a structure called pollinia. So what is actually pollinia? The all microspores held together in microsporangium of the anther. Microsporangium or anther. Okay. So these types of structure you can see in higher plants, like example calotropis. Okay. So calotropis, they have this kind of structure. This is the mass of all microspores. All the microspores are held together and they form pollinia. This is another pollinia, and this is clip-like structure present on this pollinia, which is corpusculum, which is called corpusculum. And this from this pollinia, a filamentous-like structure which is connected with corpusculum, it is called retinaculum. So in this pollinia, all microspores are held together, and this pollinia connected with the corpusculum with a filamentous structure, that is called retinaculum, and corpusculum, which is something like a clip-like structure. So these kinds of structures are present in higher plants like calotropis. Okay. In next class we will study about transverse section of anther and what are the anther wall layers and what are the importance about all these anther wall layers. We will study in next class.